Well, we are again here with marine and freshwater microbiology. As you remember, we were talking about microbial examination of water, and we will talk today about membrane filter method. In microbial examination of water, we have studied that how we can check the water quality because most of the microorganisms which are present in water, all microbial analysis, identification, and to find their presence is just very difficult, expensive, and long time taking procedure. Therefore, we have selected one idea that is indicator microorganism concept. And as an indicator microorganism, we have selected fecal coliform, which belongs from Enterobacteriaceae, and these are present in numerous number, and their presence indicates either this water is polluted with fecal or not. If it is fecally polluted, it means it contains every type of microorganism which can harm at their use, at their transfer from environment to a healthy individual. So we will talk about the membrane filter method. What is the membrane filter method? This is one technique by which we can analyze the water. We are analyzing the water for microbiology, that microorganisms, coliform or fecal coliform microorganisms are present in water or not. So for that we are using membrane filter method. Filter water through a 0.45 micrometer membrane filter. What we are doing, we are taking a filtration assembly where we are fixing 0.45 micrometer filter, or we are keeping a smaller pore size membrane. We also can put over there that may be 0.25, 0.2, etc. Which type of membrane is desired, we can put in filtration assembly. And what we are doing, we are putting water from upper side and sucking from downside, and water will pass from the membrane. What will be in the result of passage of water from the membrane, all microorganisms which are present in water sample will be trapped in filter membrane, and only water molecules will pass. So if microorganisms are present in water sample, all microbes will be trapped in filter membrane. Uh, so what we will do? We will collect this membrane because all microorganisms which were present in analyzed water sample has been trapped in membrane. We will put, we will collect that membrane, and we will place membrane on selective media. If you want to go for total coliform, we can put this membrane on Macon key agar plate. If you want to go for specific microorganism, suppose E. coli, which is a member of Enterobacteriaceae and indicator of the human feces contamination, animal feces contamination, we will put it on Macon key agar, which we had done in our routine lab, and we will incubate it at 35 degrees C for total coliform. And if you want to check fecal coliform, we must have to incubate in this plate after keeping filter membrane at 45, 44 to 45 degrees C for fecal coliform. And after 24 hours or 36 hours, we can count the colonies, that how much coliform is present in a reasonable size of water which has been filtered from the membrane. So this is called membrane filter method. So it is very easy, it is not difficult. Just we have to take filtration assembly. We have to keep a desired and required filter membrane in assembly, from where we have to pass the required size of water volume. Suppose if you want to process, if you want to check 100 ml, we have to pass 100 ml from that filter membrane. If we want to check 500 ml, we have to pass 500 ml water from that membrane, and we have to collect that membrane and have to keep on selective media. If we want to do for the general coliform, we can keep it on the concave. If you want to for the selective and specific microorganism, we can keep it on uh, eosine methylene blue EMB, and we have to incubate it for 24 to 46 hours. If you want to do it for total coliform, we have to incubate it at 35 degrees C to 37 degrees C. If you want to eat for fecal coliform, we can incubate it for at 44.5 degrees C for 24 to 46 hours. Then we can count the colonies. And another technique that is the MPN method, multiple tube fermentation method. In this method, what we are doing, we are serial dilution to extraction. We are taking samples, water sample, and we are taking uh, three set of test tubes. Three set of test tubes. In one set, we are keeping 10 ml. We are adding 10 ml of the sample. In another set, we are adding zero point 0.1 ml of the sample. And in third set, we are inoculating 1 ml of the sample. Inoculate multitube 5 or 10 of media with across the increasing series of dilution. Suppose in first test tube set where lactose broth is present in all sets of test tubes, and lowest concentration of sample we are adding zero point 0.1 ml of the sample in 3 test tubes, 1 ml of sample in 3 test tubes, and 10 ml of sample in 3 test tubes. And all 3 sets we are incubating for 24 hours with placing Durham tube, inverted Durham tube. Incubating it 35 degrees C for total coliform, and for enteric coliform or fecal coliform 45 degrees C. And we are next day we will count the positive growth tubes where acid and gas is present. Those will be positive, because if coliform is present, they have ability to produce lactic acid. In presence of acid, uh, the color of the test tube will be changed, and due to acid, due to lactose fermentation, acid will be produced. Nextly, gas will be produced, which will be trapped in the Durham tube, and we can indicate, we can check this thing uh, in test tubes physically. Use most probable number MPN table to estimate the density, and we will perform this practical in lab when physical classes will be resumed after some time. And another thing which is for field, that is enzyme substrate or chromogenic substrate method, which is very specific, very fast, and for field experimenters, if we want to go in a field and we want to check a water reservoir, or this water is microbially or fecally contaminated or not, so we can use enzyme substrate chromogenic substrate method. Used with the presence, absence, the multiple tube methods are quantitrace. Total coliform have the enzyme. We must be clear. Total coliform have an enzyme that is called beta D galactosidase. Coliform have this enzyme. The name of that enzyme is beta D galactosidase. And what does this enzyme, which hydrolyzes the orthonitrophenyl beta D galactopyranoside ONPG. So this enzyme beta galactosidase have ability to hydrolyze the ONPG. So what will be when if coliform are present there, they will produce this beta D galactosidase enzyme. If beta D galactosidase enzyme will be produced by coliform, these will hydrolyze ONPG and the color of the medium will be yellow when hydrolyzed. If coliform are not present, means there is no production of the beta T galactosidase and there will be no hydrolysis of the ONPG and there will be no change of color. Similarly, if you want to check the E. coli has the enzyme beta glucuronidase, beta glucuronidase, which hydrolyzes MUG, 4-methyl mobilifried beta glucuronide, MUG. And what will be fluorescence when hydrolyzed? It will give a color at hydrolyze. If E. coli is present, that will produce beta glucuronidase, which will react on MUG and will raise and will produce some fluorescence at hydrolysis. Like this. This one is ONGP for total coliform. If we will add water sample on this media, on this trace, which have ONPG and the enzyme which is present in water samples, it will be added over here. Color of this will be yellow. If E. coli is present, it will be like this. It will be changed. So these were two techniques. One was, if you will see here, these are three techniques which we have studied: MPN, membrane filter technique, MPN, and enzyme-based technique. So enzyme-based technique can be used for the field analysis of the water. When we are going in field and we are finding some water reservoir, if you want to check, we have to keep this type of media with us where we can add 1 drop of the water in that medium, which are available in pouch forms, and mostly NGOs are using such type of technique. We will add the water sample over here. We will keep it for few minutes. If coliform is present, the color of the media will be changed, and we can compare it with the control. Okay, this is control. There, here we have added water sample. Its color has been changed. If color has been changed, means coliform is present. If color is not changed, there are no microorganism or no coliform are present. So So, nextly we have to talk that there are various standards because world is very conscious about the water quality, because water quality, if water quality is good, uh, the health of the cities and the health of their public is good. If water quality is not good, the health of their public not will be good. So simultaneously, in result of that, consequently, what will be? If water quality is not good, it will increase disease burden on society. It will increase social pressure. It will increase economical burden, and consequently, the country will be in problem. So we are talking here Georgia EPD fecal coliform standard for water contact activity. So this is the geometric mean, GM. GM means Y1, Y2, Y3, and Y4 into 1/4. 4. At least 4 samples we have to collect for water analysis over a 3 days period. After every 30 days period, we have to collect 4 water samples to give an opinion about a water reservoir, and at least 20 hours apart. So, how we can calculate the water standard for that? We have to calculate, we have to count the geometric mean. For geometric mean is equal to that we have to collect 4 water samples, and our a 30 days period, in 30 days we have to collect 4 water samples and at least 24 hours apart from each other. So fishing and drinking fecal coliform is standard. So May to October, what should be? Geometric mean not to exceed 200 MPN per 100 ml, May to October, and no individual sample exceeding 4000 MPN per 100 ml, means 4000 microorganisms per ml not should be present in any sample. But geometric mean not should exceed 200 MPN per 100 ml. And November to April, GM not exceeding 1000 MPN per 100 ml. So number of microorganisms per 100 ml should not exceed from 1000. No individual sample exceeding 4000 MPN per 100 ml. So these are the standards. May to October and November to April. Environmental protection agencies for E. coli criteria, illness rate per 1000, and geometric mean per 100 ml, and single symbol per 100 ml. So illness rate 8 per 1000, uh, uh, 8 people from 1000 people maybe will be suffer. If what will be? If geometric mean is 126 MPN per 100 ml, and per sample maybe 235 MPN per 100 ml. Similarly, if geometric mean increases, the illness rate is also increasing, and single simple microbial load per 100 ml also increasing. So what happens in all consequences, the illness rate is increasing. Suppose if geometric mean is 206, illness rate is 9. If per sample microbial load is 300 MPN per 100 ml, so it will increase. And it is for E. coli. Look out at that, dirty birds, because up to this we were thinking that only humans are responsible to contaminate the water bodies, but here are many other creatures are also responsible for the contamination of the water, surface water. These are geese, gulls, and birds, mostly birds. They are also living in freshwater bodies. These are droplets, these are producing droplets, drops, and feces that is going into the water bodies, nearby water bodies. And they are contributing in huge number of the coliform and E. coli contamination. So, worst case, gull and goose dropping weight per gram. Ring-billed gull, it gives 0.45 gram weight of droplet, and number of microorganism is coliform is 1.52 into 10 is the power 9, and CFU of the coliform fecal coliform load is 8.21 into 10 is the power 8. So one bird can contribute huge number of microorganisms and coliform to water bodies. And if you will see this one, its weight is too huge, 9.98. and it is contributing too much, 2.47 into 10 to the power 7 microorganism to water bodies, CFU per gram, and CFU of this is 2.41 into 10 to the power 8. It means the bird fishes are also contributing in water contamination. They are uh, dropping, they are releasing their feces in water bodies, and their feces contain huge number of microorganisms, uh, and these microorganisms are contaminating to water bodies automatically, and they have to affect the human health. So maybe up to this we are keeping here, and in next session we will talk uh, ahead of it. Thank you. Have a nice time, please.